Hi, this is Dr. Randy Papke, and I'm here with a quick tutorial on how to complete the table for Lab 4 in your lab manual and your online labs. So let's just start out with a reminder that we are trying to look at a sequence of DNA, and you can tell it's DNA because it has C, A, G's, and T's. And we're trying to make an RNA sequence. In other words, we're trying to go from DNA to RNA. That process is called transcription. Also remember that when you're looking at DNA, C's go with G's in that complementary base pairing fashion, and A's go with T's, and the reverse is also true. G's go with C's and T's go with A's in DNA. But when DNA makes RNA through the process of transcription, if you have a C in DNA, you do in fact have a G in RNA. And if you have a G in DNA, you have a C in RNA. That's the transcription process. C complementary base pairs with G and G with C. If you have a T in DNA, you do in fact do an A in RNA. But if you have an A in DNA, you actually use a U, and U stands for uracil, and that's what gets put in the RNA. So we're going to only do the first 3 nucleotides, 1, 2, and 3 nucleotides of our DNA sequence for the purposes of this tutorial. And so we have a C here in our DNA, and of course that goes with a G in the mRNA. If I have an A, that goes with a U in the RNA sequence, in the transcription process. And G's, of course, go with C's. So we have now our first 3 letters. The 3 letters in a row make a codon. And what we're going to do is the translation process right now, where we're going to go from a codon to an amino acid. And you can see that these 3 in here are only going to make 1 amino acid. So we're going to have 1 amino acid put in this box right here. And in order to do that, we need a table of codons. So that's what we see right here is a table of codons. And with this table of codons, we're going to try to find the codon that we just looked up, which was GUC. And I'm just writing that there so that we don't forget what our codon was. The way that you read a table of codon is there's the first base, the second base, and the third base in the codon. And so the first base in our codon is G, and here is the letter G. So we know that our codon must be in this entire row somewhere. We can narrow it down where by looking for the second letter. The second letter could be any of these 3, but it happens to be U. So now we know it's in this row and this column, so it must be in this box. And we can find GUC right there, and we can see that it codes for valine, V-A-L, and that's just the word valine, right? It's just short for valine, val. Also notice that if we look at this table of codons, we can see that there were 3 other codons that could also have coded for val. And so that kind of repetition in the genetic code is very normal. You can see that happens quite a few times. As you finish this project, as you finish going through and writing down all of your mRNA sequence and then your amino acid sequence, you'll have a completed amino acid sequence. You'll do the same thing for the mutated sequence. In this case, we're talking about sickle cell disease, which is a mutated form of hemoglobin. So there are a few mutations in here in the DNA. Mutations are always found in the DNA. Sometimes they have consequences that affect the amino acid sequence, and sometimes they don't. It's your job as you go through this project to figure out where the mutations are, and you'll identify those mutations based on nucleotide number. So this is nucleotide 3 in normal, this is nucleotide 3 in sickle cell. There's no mutation there, right? So that's kind of what you'll be doing. I hope this tutorial is very helpful to you as you finish up Lab 4 in Bio 100.